Good afternoon, everybody, and welcome to the first interdisciplinary lecture series, seminar of this interdisciplinary lecture series of this network. Um, we are very happy that the first lecture is going to be delivered by Carlos Marques, uh, from the École Normale Supérieure of Lyon, where he chaired the lab of chemistry. And he's a super expert in lipid membranes, and I think is one of the best persons that can introduce you to this topic. And I think that you can get his CV in the web. I mean, he was in several very prestigious laboratories all over his scientific career, and he also, I think, is a very important contribution. Is the book that he wrote with Rumi Dimova about the giant vesicle book. I think is a masterpiece. And without more introduction, I think I will leave the floor to Carlos to give the lecture.
Okay, Herman, thank you so much for these kind words. So you all know the four pillars of life, the sugars, the proteins, the genomic information, and the lipids. And let me start by proposing that from those four pillars, lipids have been always, or for many people, considered as quite dull and not interesting at all. And why was that? Because there is no very much defined molecular structure like with the proteins or with the DNA that would convey information, for instance. There is no high specificity like the carbohydrates. So for a long time, they were considered, as Ole Morrison wrote, as a structureless fatty material that basically was oiling the mechanics of life. And I'm sure that after your PhD, that will be dealing with lipids and proteins and many other things, you will think otherwise about lipids, which actually started to be important or understood later than the other ones. One mark of that, you might say, is that the words associated with the proteins, the DNA, and the lipids, which are genomics, proteomics, and lipidomics, appeared for the first time in this sequence, and you see that the lipidomics are the ones that are the youngest ones. So in that sense, dealing with lipids is quite a young science, at least dealing with lipids from the point of view of saying that they are really important and their organization needs to be understood for understanding life and many other things. A person that I would recommend reading, and if you have never read this book, I strongly recommend you to do so, is the book by Ole Morrison, which founded at the University of South Denmark, a big group working on the physics of membranes. And he wrote this wonderful book where he discusses why lipids are important for life. And lipids, of course, they are associated with fat, and he called it very wisely life as a matter of fat. In this edition, there was a contribution by Luis Bagatoli, a big name in the science of lipids that unfortunately deceased. He also translated this into Spanish. So if Spanish is your preferred language, you can buy this book translated by Luis Bagatoli. So what are lipids? I like to introduce them like this. You all know what this tree here is. A palm tree, is one of the palm trees, and these ones, they gave this kind of dates, and from these dates, if you cut it, you see there is some content inside, the central kernel and the mesocarp around it, and if you press it, from the mesocarp comes an oil, and this oil actually is palm oil that is sold all over the world by more than 70 million tons a year. So this is one of the most sold products from natural origin. And what it consists of, it's of this molecule mainly, not exclusively, it's not 100% made out of this triglyceride, but most of it is tripalmitoyl glycerol. And what tripalmitoyl glycerol has, of course, is three chains connected by a glycerol here in the middle. And if you look at it carefully, and perhaps you already knew this or you already noticed this, but it's not enough to emphasize that in lipids, most of the atoms that we have are very low atomic number atoms. There are hydrogen, of course, 1 electron, a carbon with 6 electrons, and oxygen with 8 electrons. And why this is important is because if you look for the interactions between, say, radiation or other things with this material, that is going to be determined, for instance, if it's X-ray, by the amount of electrons, if it's neutrons with the amount of neutrons. And so we are dealing here with very low numbers, and it's good to keep that in mind. An important point to make about these triglycerides is that they are a kind of Lego. They are assembled from a glycerol that you see here. And it's important to know that the glycerol, although we are able to put it on a plane like this, so it's a kind of flat molecule, of course it has some 3D structure. And you see that because of that, there is some stereochemistry involved, and you can name the carbons on it like 1, 2, 3, if you start from one end and leave the hydrogen in the middle to your right. So this makes 1, 2, 3. And so it's non-chiral molecule, but as soon as you attach to a molecule, it becomes chiral because precisely of this point here in 2. This glycerol is attached to 3 fatty acid chains. Why do we say there are fatty acids? Because at the end, indeed, you have a carboxylic acid with 2 oxygens and hydrogen here and the carbon, and is the esterification of these 2 things that gives the glyceride which is in there. Now, this is called palmitic acid. It has 16 carbons, and if you look at these other 3, the ones that came not from the tropical area, but rather from around the Mediterranean, recognized as an olive tree. So the olive tree gives olives, as we know, at least ones that live around here. And if you make a paste out of olive and put it to press, you can extract an oil as well, like for the palm oil. It's not sold for so many tons. It's maybe 10 times less than palm oil, but it's still a significant amount. And it looks very much like the other one. Now, this one here is called trialloyl glycerol. Now we have 18 carbons and a feature in the center, which is an unsaturation. And so it looks very much like the other one I was talking about, except for the unsaturations and a 2 carbon difference. So visually even it looks different. And it also has hydrogen carbons and oxygens. And as I was saying, there is a different feature is in the middle, which is an unsaturation. You see, not only it makes it more difficult to draw, but it also gives the 3-dimensional features of the molecule different because it needs to bend, but it's also an acid, a carboxylic acid. This one is oleic acid. And when esterified with the glycerol, it can give a triglyceride. So we have here already the basis for the lipids. Let's now change scale. I'm not talking about trees still, but I'm still in the plant realm. These are algae. These are green algae called Chlamydomonas, and people talked a little bit about this because wonderful cryo-electron microscopy images were made out of these cells not so long time ago, which for the first time um, could display the internal separation of the cell based on the lipids to an extent that had not been achieved before. So this is quite a wonderful three-dimensional representation of these walls inside that single cell that are made out of lipids. And they are made out of lipids that look like this one. It's not exclusively this one, but this is another lipid. You'll recognize here attached to the glycerol, oleic acid, as you have seen before, a palmitic as well. So eventually these cells have molecules that resemble a lot like the ones of the tree, of the dates and of the olives. But here to the right, on the position 3 actually of the glycerol, you recognize a few new elements that are written here as PC, which stand for phosphatidylcholine. And now we add two more atoms to our collection of atoms, and this is be pretty much it for lipids and phospholipids. We have, of course, nitrogen, which has 7 electrons, and phosphorus, which is the heaviest of the elements in lipids, which has 15 electrons and 15 protons, of course, and 16 neutrons. Of course, they are variants of these with plus or minus neutrons, but this is the most common one. So the new specificity of this part that phospholipids have is that contrary to the tails that we are discussing before, which of course are oily and so are hydrophobic, this head here is well soluble in water, and so it's hydrophilic. So now we have a molecule that is amphiphile. It has an hydrophobic part and an hydrophilic part, and this will give it, of course, properties that are of interest to us. So I said that molecules are like POPC, the ones that make the membranes in that cell. Are they all POPC? Of course, we know they are not all POPC, and as a matter of fact, there is an amazing variety of molecules that one can call lipids. For instance, you can have the same tails, and again here we have palmitoyl, oloyl, and we can, with the same tails, we can attach different heads. Here it's called phosphocholine, I showed it before. Here is a different one, still has a phosphorus and a nitrogen, is ethanolamine. Still a different one that also has a phosphorus and a nitrogen, but a few more oxygens. It's made of an amino acid, is serine. And this one here doesn't have nitrogen, it has the phosphorus, is phosphatidyl glycerol. So you see, same tails, we have different heads. So already in here, we have quite a variety of molecules that constitute the lipids. And an important point to make about what these different heads provide, of course, they have different sizes because they have different amounts of atoms and so on. But also they have different charges, and this is quite important. These ones here in the top, the phosphatidylcholine and phosphatidylethanolamine, they are zwitterionic, meaning that they are indeed charged, but they have a negative charge and a positive charge that are not dissociated. So they are overall neutral with a dipole here. And otherwise, all other molecules under physiological conditions, they are negatively charged. So there are no naturally occurring positively charged lipids, but of course one can make positively charged lipids by synthesizing the right lipids. So keep that in mind, no positively charged lipids occurring naturally. So as we can have the same tails and different heads, we can have the same heads and different tails, of course. So if I take phosphocholine, I can have an enormous amount of different tails. We already saw the olive oil and the palm oil variety. Here is a lipid with two oleic acid tails. The OPC is a star with lipid scientists because it stays in a fluid state that, as we'll see, it's important. And there is a new one, stearoyl. So it's like the palmitoyl, but it has 18 carbons and no unsaturation. Stearis comes from the Greek and means ox fat, actually. So you find lots of these big chains saturated in animal grease, actually. Again, a story of fat. You can have the same tails and the same heads and still have different lipids. This is, I think, important to restate because for some applications might be important. So here is our POPC, the ones that we opened the lesson with the palmitoyl, with the oleoyl. As I told you, the glycerol has a 1, 2, 3 carbons which are different that can be indexed. And so if I attach the oleoyl chain to position 1 and the palmitoyl to position 2, I get a lipid that has exactly the same amount of atoms and the same atoms, but which is a different lipid, and it's called OPPC instead of POPC. And it's different lipid because it's a stereoisomer of the first molecule. So it has actually different properties. It has, of course, different chiral properties because the light is not going to interact the same with this stereocenter in the middle of the glycerol. But interestingly, these differences in attachment of the different chains also change other properties. It changed the thermodynamic properties, it changed the interaction. For instance, this is important in determining which best anesthesia molecule can be important to make you sleep during surgery, for instance. So same tail, same head, but still different lipids depending on how the tails are attached to, to the glycerol. We have lipids which don't have two tails, and here is a typical example that is very abundant in mitochondria, for instance. It's called cardiolipin, and it's a kind of POPC, POPG with the head of glycerol, which is attached by a glycerol here. And you can guess that the way that the cells do this is first by making the lipids and attaching this little glycerol here at the end. And as a matter of fact, if you take bacteria and feed them with a different alcohol, like mannitol, which is like a glycerol but a little bit longer, they end up inserting mannitol here, and you can have a sort of cardiolipin which is completely modified and actually makes the bacteria much more susceptible to be attacked by light treatment, for instance. So the way these things are built is important. And they are lipids which are usually called lysolipids, which have been stripped of one of the chains. They are called lysolipids, and they are important also for other reasons. But you see that in a lysolipid, you have one chain attached in position 1 usually, then this space here is free and the head is the same. So actually to do chemistry building new lipids, this is quite a nice precursor, and many companies that make lipids for you, they start from here and then attach to this other position whatever chain is available and you need for your research. So phospholipids are not all the lipids that one can talk about. Not everybody has a glycerol. For instance, here we have the same head is a choline here, but we don't have a glycerol group in here. This is a sphingolipid, so it's different. It exists a lot in the brain, for instance. And we have the star of the lipids in the mammalian cells, which is cholesterol, because we know that without cholesterol, the cells cannot function. It's also a lipid, but you see that it's completely different from the other ones, and they are full groups of research that focus actually on just understanding cholesterol. So we could say, okay, that's enough for a variety, but keep in mind that I have only touched the tip of the iceberg, and that if you dip into the lipid variety, you'll be amazed by how much there are. For instance, if you look at archaea, which are these very old unicellular organisms that can live also in extreme conditions, they can be extremophiles. They have lipids, for instance. Lipids are lipids that go across the membrane. So if you just make a membrane of a lipid, you can't really tell it's a bilayer because the chains are connected. And now you have this methylene group 0 on the side. So we are not talking about the alkyl chains as before. So this, you see, this is quite different. Here there is even a different group. So these are lipids from some species. And in your body, when you are being oxidized because you age and you expose yourself to light and so on, you can also have your lipids acquiring an hydroperoxidase group here in the middle. And so these chains now are not just as I described before, but they can have groups attached here in the middle. And of course, this changes the membrane properties a lot. So keep in mind that the variety of lipid is enormous. It's out there for you to discover. And the way perhaps of going about it is to try to start to be aware where each of the lipids are. And there are a number of publications for this. If you look in the book that I'll introduce later, but that Herman already mentioned, the Giant Vesicle book, there is a chapter where this is described. I took the pictures from there. For instance, there are differences between the lipid composition in the plasma membrane, which is the membrane outside the cells, and in the other lipids of the cell. I'm not going to spend much time describing this, but be aware that different lipids are at different place, and there is a reason for that. There is a metabolic reason for that. There are different lipids in different organelles, for instance. I was saying that there is lots of cardiolipin in mitochondria, but you can look at this kind of picture and try to make yourself aware of which variety of lipids exists in which part of the cells, if this is of interest to you. Okay. So lipids assembled then as lipid membranes, as we saw. I was showing this electron micrograph. But so what is a membrane? Well, a membrane, as you already know for sure, it's usually a bilayer where the heads of the lipid go to the outside and the tail stay inside, screened from the water. And this, of course, here is just a very rough sketch. If you make a computer simulation with an atomic representation of lipids, it looks a little more like this. So there is more disorder, and here there is disorder because you are at high temperature and the chains wiggle a lot. And the other thing that we should keep in mind is that this little thin piece of oil here is really thin. It's only 5 nanometers, which is very small compared to the other dimensions that you know. So it is a nanoscopic structure, but despite of that, it holds all the organization of life. So a pretty impressive achievement for this self-assembled piece of material, of oil, basically. So they assembled as lipid bilayers, but it's perhaps worth saying a word about the different kinds of assemblies that we can find in nature or in the materials. And if I ask the question, is because the answer is no, of course. They don't all self-assemble as bilayers. You all heard about micelles. Here is a computer simulation of a kind of amphiphile molecule that that assembles as a spherical object that is called a micelle. This is a kind of historic picture because it's amongst the first predictions of a now very popular simulation tool, which is called the Martini force field that was developed by by Marink in Groningen and is now widely used in the world. I happened to meet him from the beginning of his activity, and he kindly gave me a few pictures of the by then emerging simulation method. You can also tune a little bit the molecules and get cylinders, get warm like micelles, and this is another prediction from Jan Sievert. But keep in mind that lipids can make many other structures. It's not only spheres and cylinders. Lipids can make bicontinuous structures like the ones that are here. And if you break these structures, you can make small bits that are still organized inside. This is called a cubosome because inside there is a cubic phase made out of monoline. So monoline is olive oil where you took out two of the fatty chains and just kept one. This is a material that is used for many purposes, but it was first studied by this person that I encourage you to discover more about, Vittorio Luzzati. Vittorio Luzzati was an Italian-born scientist that worked in France, first in Strasbourg and then in Paris. And he understood in the 60s that behind this simple molecule, one could find a wonderful variety of liquid crystalline structures. This is a person that went through Second World War in difficult conditions and started being a scientist after that. He tells a little bit about his life in an interview of about 1 hour long that you can find in the Polo repository of the University of Cambridge. And if you type interview of Vittorio Luzzati, I think you'll have a wonderful time listening to this guy by the end of his life, giving this interview. And here the comparison of two structures, one very very well known, is the liposome on the left. You see the bilayer structure of the liposome. So this is a cryo-electron microscopy. And here to the right, it's something that resembles, but it's not quite the same. And if we were interacting in a room, I would ask you if you ever have seen image like this on the right. But let me just tell you, since we are at a distance, that many of us actually had these little structures in our bodies during COVID, because this is a mRNA COVID vaccine, which was developed and able to be developed due to the lipid nanoparticles that carry the RNA for delivery. And so it looks like this. It's of course a more complex structure that is the object of much research these days. So why different lipids do different structures? You probably already heard about this, but let me tell you about the packing parameter that was first developed or understood by Jacob Izraachvili and his colleagues. And the idea is very simple, is that if you have a big head then and a little volume of the tail, then you want to have a big space outside, and this is going to force spherical geometries, and all the way down to cylinders where you'd have something like planar bilayers. And such a simple concept actually captures a lot of the science between the geometry of the organization of these self-assembled objects. And this is certainly the second book that I recommend to have as a book if you want to understand the molecular basis behind the self-assembly. Jacob Izraachvili, this is a few years ago, he wrote an immensely popular book that has helped many scientists to acquire intuition about the specifics of self-assembly. And this is not, this packing parameter concept is not that concept. It's actually still being used here in computer simulations to try to rationalize what happens with different kinds of lipids when they pack together. This is a paper from 2022 discussing in terms of this simple parameter what kind of assembled you get from these common lipids that we talked about, POPC, DPPC, sphingomyelin, and so on. So we have been talking about lipid membranes. Let me perhaps stress a point that is perhaps not stressed enough about for how long we have known membranes. What you are looking at here is perhaps the oldest written piece of literary composition, is the Epic of Gilgamesh, and this appeared perhaps 2000 years before our era. And so literature is very old, science is very old, and how old is our knowledge of membranes? For this, I'm going to borrow something that we have in the membrane book, which is a little timeline here. Let me go through it, because perhaps we don't realize that it's not so long time ago that we started being able to see what is happening at the microscopic level. This is one of the first microscopes that was developed a little bit between the 1700s, and with which people saw cork. That's why things started being called cells, is because of these images seen under that simple microscope. And we only started to understand that the cells had a kind of boundary about 220 years ago. And from there on, it took a while. It took about 20 years to understand that that boundary was actually a bilayer. And Langmuir and other people, by working on water-air interface, they contributed to this a lot. But one needed to wait until after the war, which is in the 1950s, to have the first electron microscope images of bilayers, and to see that indeed the bilayer is a bilayer, is the addition of two monolayers, one against each other. And we started being able to assemble these lipids as giant vesicles that we now can look at under an optical microscope not so long time ago, maybe in the 60s or in the 70s. So just to give an idea that we are talking about a science that is relatively new. Talking about giant vesicles, with Rumi, we put together the contribution of many colleagues and friends that made a book where you have the fundamentals of understanding lipids from the point of view of these giant objects, giant from the point of view of the microscopic dimensions. So we have been talking about the molecular structure a lot of the lipids, and it was perhaps my choice to start this series of lectures like that. But it's true that the molecular structure determines everything else. So it's important to know that what are the connections between these things. And so if I ask what are the properties of lipid membranes that are influenced by the structure of the molecules, I'd say everyone. Everything is determined at that level, and being able to understand what is the connection is something that is part of what we need to understand when we work with lipids. And here is an important part of those properties, thermodynamics. I think this is the one we always should start from. It turns out that when you change temperature of the bilayers, they go through different states. At high temperature, they are in the so-called fluid state. Then they go from the fluid to the gel transition if you lower the temperature, and they can even go into a more crystallized format for some of the lipids at lower temperatures. So the fluid state, it's very disordered. It looks like a night dance club, while the gel is more like not so strict army, and the crystal, of course, is very, very organized. And this has important consequences for the mobilities of the lipids, and therefore for all the possibility of reactions and rearrangements and so on. For the rigidity, the gel phase and the crystalline phase are much more rigid than the fluid phase. And for the functioning of cells, the fluid phase is very important. And it's also important because then it determines how the cell functions, the temperature at which these lipids can go from fluid to gel. Um, of course, this is a horrible table because it's too big, but I just want you to focus on the fact that the numbers at which of the temperatures at which the transition takes place, they vary widely. They go from -70 to up 80. So you see, you can be all over. So depending on the lipid structure that you have, depending on the number of unsaturations, the number of carbons and on the heads, you can have very different temperatures. For instance, here is the temperature of the transition, the fluid gel transition for PC, PS, and P lipids as a function of number of carbons. And you go for the PC heads from 0, around 12 carbons, to 80 when you have 24 carbons. So there is a high variety, and this is highly significant. A good way of entering the field of understanding the thermodynamics of lipids is to read a few of the chapters of this book by Thomas Heinburg, one of the best experts in the field, because all of this is related to the molecular behavior of the molecules, and you can relate these to that. And as a book that you need probably to understand the structure and have all the numbers, you might want to have the Derek Marsh not very far, because all the numbers you want to know about lipids are inside this maybe 1,000 pages or more collection of properties by Derek. So if you put together two lipids that have different temperature of phase transitions, of course, they are going to phase separate because they are not happening together. One is more solid, other is more fluid, and they make phases, lateral phases that we call domains. Cholesterol also can phase separate. You can make phases which are rich in cholesterol and other ones that are poor at given temperature. And if you make ternary binary mixtures, this is actually relevant for protein activation, and maybe some of you already have seen this movie, which is a hit on the web with students in biophysics. It's called The Inner Life of a Cell, and what it shows is that when proteins need to be activated, some of them, they are actually activated by the process of phase separation that is induced by lipids. So the lateral phase separation of saturated and unsaturated lipids and cholesterol brings protein together, then are activated and can do their job, whatever that job is. So this phase separation in lipid bilayers has been the topic of lots of research these past years, and I'm sure they will be discussed in the context of our network because they are important certainly for the interactions with the condensates. They are usually described, the ternary mixtures, in this kind of diagrams. So you make diagrams and look at how the vesicles phase separate with some domains that are more fluid than others, or you can have fluid-fluids phase separation. This is a wonderful domain of research that has been rather explored already for most well-known lipids, but not in the context so much of the interaction with other molecules such as proteins. So I have 2 minutes, so I'm not sure I'm going to indulge into spending much time discussing how does one prepare membrane systems. And so perhaps I'm going to stop sharing here and just say a few words in the end. So one should not at all be restricted to the discussions I chose today, and I could not talk very much about system preparation, which is important, but you'll have it on my on my slides. We need also to wonder about what techniques are better to studying the lipid membranes, and there are many available. So this might be perhaps the subject of some other lecture in this series. And of course, there are many ways of helping experimentalists during the experiments. There is theory, which helps you sometimes understand your experiments and many times to plan them. And there are these days computer simulations, which are also very important in the sense that you can interact with groups during computer simulations and try to use that to understand better your system, or if you don't, to challenge your computer simulation colleagues to develop research in that direction. And with this, I think I spent 45 minutes more or less, so I'm going to stop here